Hi, welcome back, guys. So we made it to the final, and hopefully you've gotten a lot out of the course, and you've kind of made some connections about the real world and biology, and it really made a foundation for maybe when you are reading something that has some kind of biological background to it. It kind of makes a little bit more sense of what's really going on, what's the underlying thing there or concept there. So um, when we're talking about, I just want to go over some things for the final exam. Obviously, the lectures are pre-recorded. If you do not remember something, if you're having trouble with something, you can go back and look at them to refresh your memory. You can email me if you have an issue or a question. But this is going to serve as kind of a quick overview of what we learned this semester. Obviously, I'm not going to go completely into everything again, but just to try to give an overview. This is to help. So remember, we are in a biology course. So biology, when we talk about biology, well, what is biology? Biology is the study of living things. So there's many different types of sciences, and when we're talking about the different types of sciences, anything that's going to be involving living things, we're going to be talking about biology. So when we talk about chemistry, we're talking about molecules, the way that they come together, they bond, and we're talking about earth science or geology. You're talking about rocks or the physical non-living things on Earth. But when we're talking about biology, we're talking about the study of living things. And in any science course, we like to talk about the scientific method. And in this course, we like to keep it very basic and talk about just the steps. So in the real world, it's a little bit more convoluted, and you kind of have to go from one step and go back two steps, and then go to the next step, and maybe go back to a step. But we like to just think of it as an order that we have, and usually we're going to have a problem, we're going to have a question, then we're going to possibly do some research, maybe do create some kind of prediction about something, and we're going to formulate a hypothesis. Once we formulate a hypothesis, we have to come up with a way of conducting an experiment to test that hypothesis. And what we like to do is we like to do controlled experiments where we are going to be able to control all variables and only change one variable at a time. So we're only changing one variable. I always like to use the example of plants. So when we're talking about doing an experiment with plants, right, we want to test something like does fertilization for using fertilizer really work and help to grow plants. So we can take two sets of plants and we want everything to be the same except the idea of fertilizer or no fertilizer. So we're going to use the same soil, we're going to use the same amount of water each day, we're going to use the same environment, same amount of sunlight, same size container that we're going to keep them in. Everything is the same type of plant. Everything's going to stay the same except one group is going to get fertilizer, the other group is not going to get fertilizer. And ultimately, what is the scientific method? It's a way of testing a hypothesis. And this hypothesis, when we're talking about testing a hypothesis, this is a process, scientific method, the process of looking at testable and measurable events being observed. So we're observing testable and measurable events. And when we start getting into biology, we like to first start talking about biochem or chemistry, really. And when we talk about chemistry, remember we're talking about what makes up an atom. You have your protons, you have your neutrons in the nucleus, and then on the outside you have these electrons. And we like to talk about bonding. And with bonding, we are going to have ionic bonds, and we're going to have covalent bonds. And in our ionic bonds, you're going to see that the electrons are going to be either donated or accepted. And with covalent bonds, the electrons are going to be shared. And that's how different molecules come together. Now, there are other types of bonds, and we can get more specific into polar, non-polar, excuse me, polar, non-polar. But when we are talking about a very basic science course, we like to just address just very simply, we have ionic bonds and we have covalent bonds. There are hydrogen bonds, which are important in DNA, which we'll get to in a moment. And then once we um, started to talk about different chemistry and the chemicals and the atoms and the way that they bond, we like to talk about organic molecules. So we can take every type of molecule and we can break them up into either them being inorganic molecules or organic molecules. And now when we're talking about organic molecules, it's not the way that you may think. When we hear organic, we often think about going to the grocery store, getting food, organic foods, and those are really the foods you want. Those are the foods that haven't been created by using GMOs or pesticides or anything like that. But when we're talking about organic in chemistry or in science, we're really talking about the idea of them having carbon. So carbon is what makes things organic. If it contains carbon along with hydrogen, then it is considered an organic molecule. There are molecules that don't have hydrogen with carbon, so they're just carbon and some other atoms. They are not considered organic. But whenever you have an organic molecule, it is going to have carbon and it's going to have hydrogen. And most of the time, it's also going to have oxygen. So when we're talking about organic molecules, we have carbon, we have hydrogen, we have oxygen, and then we have other molecules as well. But when they don't necessarily, the other ones don't have to necessarily be there. There can be other ones there, but they're going to have carbon and hydrogen, and most of the time oxygen as well. Now, when we talk about organic molecules, we're talking about 4 different types of organic molecules, and we talk about carbohydrates. And carbohydrates are sugars. So as much as people often think about carbohydrates as being bread, pastas, rice, well, yes, that's true. Your starches are carbohydrates, but carbohydrates are anything that contains sugar. So candy is technically carbohydrate, even though when you hear someone say, "Oh, I need to cut down on carbs," a lot of times they're referring to the amount of rice, potatoes, pasta that they are eating. Then you have your lipids. They are your fats, oils, even waxes. And then you're going to have your proteins, and you're going to have your nucleic acids. And proteins, there are many different types of proteins. Enzymes are one type. You have hormones are another type of proteins. And then your nucleic acids are going to be made up. Either you're going to have RNA or you're going to have DNA. Those are the 2 types of nucleic acids that we see. Now, once we moved past our organic molecules, we started talking about the cell. And a cell is really the basic unit of life. Now, what do we mean the basic unit of life? It means that anything simpler than a cell is not necessarily going to be a living thing. It can be part of a living thing. So you have different molecules, you have organelles. These are all parts of living things. But until it is a cell, it is not considered a living thing. If something is a cell, it's a living thing. If something is made up of cells, it is a living thing. So we consider anything that's a cell itself or made up of cells is a living thing, which is why we call it the basic unit of life. Now, there's different types of cells, and we like to talk about prokaryotic cells, and we like to talk about eukaryotic cells. Now, prokaryotic cells, we think of them as being very simple, and eukaryotic cells are more complex. Now, prokaryotic cells are, bacteria or prokaryotes. And then we talk about more complex things that are made up of more complex cells, we're talking about eukaryotes. So we are eukaryotic organisms. Our cells are more complex. So in prokaryotic cells, they don't have no membrane bound organelles. So they do have organelles. They're going to have ribosomes. They're going to have a cell membrane. Those are organelles, but they're not going to have chloroplast. They're not going to have mitochondria. But in eukaryotic cells, they are going to have membrane bound organelles. And now when we talk about cells and how cells are made up, how organisms are made up, we love to talk about genetics. And genetics was a major part of this course. And we like to talk about DNA. Now, DNA, when we're looking at it, we like to look at it as an unwound thing. So it's unwound, looks like a ladder when it's unwound, but in reality, if we don't unwind it, it is this twisted ladder known as a double helix. So it has this double helix shape to it, and it contains this genetic information. We use the code in order to code for specific proteins, in order to know what proteins to build. Now, what we can do is we can look at what are these things made of? How is DNA made up? Well, a segment of DNA, if you take this area right here, because we have these different segments, and what you have is you're going to have an area where there is a sugar. So we have a sugar, and then you're going to have an area which is a phosphate group. All right, so that's really what's making up the vertical section of the ladder. But the horizontal section, the rungs of the ladder, are made up of these nitrogenous, or sometimes called nitrogen containing bases. So you have these three parts. You have a sugar, which is either going to be ribose in RNA, or it's going to be deoxyribose in DNA. So you're going to have a sugar, that's one part of it. Another part is going to be a phosphate group, and then you're going to have these nitrogenous bases, which are going to be, those nitrogenous bases are going to be either your A, your T, your C, or your G. Remember, when we talked about mRNA, it does not have a T, instead it has a U, so you're going to have A, U, C, and G. And remember, when we talk about complementary base pairing, the way that they come together, we know that if one side of the ladder has an A, the other side has a T. If one side has a T, the other side has an A. One side has a C, the other side has a G. And if one side has a G, the other side has a C. And the same thing goes for RNA. If we are going to take a code of the, what happens is we can either duplicate or replicate the DNA. All right, so DNA replication involves having these rungs, and what happens is we're going to split this down, and one's going to go over here and one's going to go over here, and just by knowing, so if I take this side and this has, say, A, T, C, and this side has T, A, G, because they were paired up previously, well, now I'm going to build a new other side. So this is going to make a T, because I know A goes with T. This is going to make an A, and this is going to make a G. And the same goes with this one. So now I have two DNA molecules just from one. So by having one DNA, we can split it in half and then build off of each half, all right, or each strand. So you have these two strands, we separate them, and now we can build off them to make two completely separate DNAs. So We're not just building a whole new DNA from the old one. We're not just making a copy of it. We're splitting it in half, and now we're building off of each one. And that's with DNA replication. But when we talk about making proteins using the DNA to create traits, we're going to be talking about protein synthesis. And what this does is we take our DNA and then we are going to make our mRNA, and our mRNA is going to be sent out to the ribosomes, and then the ribosomes are going to build our protein for us. So as much as we can make new DNA from DNA, sometimes we want to make proteins. A lot of times we want to make proteins from them. We want to create a trait. That's where we see genetics really shine through to what it means for an individual, what it means for that organism, what it means for evolution. So with protein synthesis, instead of our one DNA making two DNA, we have a DNA strand, which is going to help us make an mRNA strand. And this process is called transcription. And then what we're going to do is we are going to build amino acids. We're going to put amino acids together. And this is called translation. And amino acids are the building blocks of protein. So that's how we make our proteins from our DNA. So if you're ever wondering, how is it that DNA becomes something? How does DNA have an influence on who we are, what we are? That is exactly how it happens. And when we talk about genetics, a lot of times we like to talk about biotechnology. And biotechnology is really just the idea that biology and technology come together, and we can use biology in order to move ourselves forward technologically. So as much as biology is the study of living things, we can use biology as a kind of a form of technology to help us along the way. And what we've done is it helps us to feed a growing population. So as time goes on, our population is getting bigger and bigger and bigger, and it is harder and harder to feed them, especially when you have issues with having a steady amount of food, issues with your food sources, making sure that not only we can have access to plants, but also the meat that we eat, they will have access to plants as well. So biotechnology is important in feeding a growing population, also important in treating diseases, as well as important in diagnosing genetic diseases. And what happens is we sometimes like to genetically modify organisms, GMOs, and this has a bad connotation to it. A lot of people think of it, oh, GMO, that's a bad thing. Reality, it's helped us a lot along the way as far as a population is concerned. So individuals might have issues with it, but as a population, as our population grows exponentially or has grown exponentially, GMOs were important. And what we had in GMOs was we had to create these transgenic organisms. And transgenic genic organisms are organisms with pieces of other organisms' DNA in their DNA. So what we do is we find a gene, we find a segment of DNA that we like in an organism, and we say, you know what, we want to take that from that organism, put it in this organism. So if there's something, if an organism makes a special protein and that protein helps it in some way, we want to insert it into that other organism to help it that way. So what we've done is we've inserted genes for being able to rid the organism of pesticide of pests of insects. So we're kind of inserting a gene for pesticides in that organism, and that's where we see things like BT corn. We've inserted genes from a bacteria that is able to fend off pests, and we are inserting that into the corn. So the corn has its own type of pesticide in it. Another thing we see is golden rice, and golden rice is able to make beta carotene. So by giving it that ability to make beta carotene, now people are able to eat this golden rice, and now they're able to get their beta carotene if their diet is short in beta carotene. So in certain parts of the world, people aren't getting enough beta carotene, and those people might eat a lot of rice. If they eat a lot of rice, we can give them this golden rice instead of the old type of rice, and now they are no longer deficient in beta carotene. So biotechnology has really helped along the way. Now, we see that when we're trying to diagnose genetic disorders, there are different genetic disorders. We look at things like um, short, shortened telomeres. Well, that is kind of when we look at a a chromosome. A lot of times we like to draw it as an X. It's really kind of like this. And on the tips, you have these caps, and these caps are called telomeres, and they get shorter and shorter as time goes on, and that's what uh, is going on when we are aging. But we can see that there are some people that are aging very rapidly, and those people have an early onset of these shortened telomeres, okay? And what we call it is Hutchinson Gilford progeria syndrome. So just just some examples of biotechnology and being able to use that for our advantage. We're able to diagnose genetic disorders. We're able to combat genetic disorders, combat diseases. We're able to use it to help with excuse me, crop growth and feeding our growing population. But what we want to remember is that we talked about when we talked about earlier, we're talking about chromosomes. A lot of times we brought up homologous chromosomes. And homologous chromosomes are chromosomes that have the same genes on them. They don't necessarily have the same information, which the specific type of gene or version of gene, I should say, is an allele. But they don't necessarily have the same allele, but they code for the same things. So we call those homologous chromosomes. And everyone has two of that version. So you're going to have, we have 46 chromosomes in total. You're going to have two number ones, two number twos, two number threes, all the way up to 22. So two 22s, that brings you to 44. And then you're going to have your sex chromosomes, and either you're going to have an X and an X, which makes you a female, or an X and a Y, which makes you a male. So we talk about homologous chromosomes. And where these come up is when we talk about meiosis. Now remember, with mitosis, we talk about IP-mat. Now, IP-mat, you have interphase, prophase, metaphase, anaphase, telophase. Now, in interphase, that's when we're going to see DNA synthesis happening. We're making more DNA. And in prophase, we're really condensing the DNA. And then in metaphase, you're going to see that all that DNA is going to go to the middle of the cell, and then we're going to split up that DNA and is going to, in anaphase, move away from the center to the different poles. And then in telophase, the cells are really going to separate, and in cytokinesis, that the cell membrane is going to pinch off and make two new cells. So with mitosis, you start with one cell that has the full genetic information, and we call that a 2N cell or a diploid cell. 2N is the full amount of genetic information. And we're going to replicate that, and we don't really call it a 4N cell, but we say, hey, we now have a double of the 2N, and then we're going to split that up, and when we split that up, now we have a 2N and a 2N. So this is great for if you cut yourself, you want to make new cells, you're trying to repair growth, you want more of the cells with the full genetic information in it. So our cells have the full amount of genetic information in it. If we want to make new ones for repair and growth, we want those to now have the full genetic information in it. So with mitosis, you need that interphase where you're synthesizing DNA, you're replicating that DNA, you're taking your DNA, you're making another copy of it so that in mitosis you can separate it. You need that to happen. Otherwise, your 2N cell is going to create a 1N cell and a 1N cell, two separate 1N cells. But then we have meiosis, where we want to make sex cells. We want to make sperm and egg. And what happens is we have our 2N cell, and we want to make 1N cells. So we're going to do the same thing as mitosis. We're going to create that. So we're going to have our 2N cell, and then we're going to end up making two 2N cells, just like we did in mitosis, but then they're going to separate again, and you're going to end up with a 1N cell, with four 1N cells, I should say. Okay, now what happens in meiosis, we have interphase, then we have prophase, metaphase, anaphase, telophase, and then we have, again, prophase, metaphase, anaphase, telophase. Now, in the first one, what happens is if we have a cell and we have these homologous chromosomes, right, they are going to all be lined up and say I have these are the same, these are the same, these are the same. You're going to see one's going to go one way, the other's going to go the other way. One's going to go one way, the other's going to go the other way. One's going to go one way, the other's going to go the other way. And then what happens in the next one is if you have two of them where they have these X's, which they're not really X's because we can separate them, what's going to happen is you're going to have one line go one way, one line go the other way. And what's going to happen is you're going to have these cells end up looking like they have this in it. No more X's, just lines, because we separated those X's, which are not really X's like this. They actually look like this. And we're able to separate them down the middle. So when we look at genetics and evolution, you're going to see that evolution, originally Darwin described it as descent with modification. Now, what is descent with modification? Descent with modification is as time goes on, organisms are going to have things happen to them and they are going to change. Some traits are going to be good, some traits are going to be bad. And as time goes on, as they reproduce, the traits that are going to help are going to be passed on, and traits that don't help something survive, those organisms are not going to live long enough to reproduce and pass those on. So we have different agents of evolution or agents of evolutionary change. And, there's different ones. Natural selection is a big one. The idea that nature is going to pick and choose which organisms are fit and which ones are not. It's really the environment is saying this trait helps them live, this trait doesn't, and they're not going to live long enough to reproduce. So what you really want to do is live long enough to reproduce. The more offspring you can make, and the more fit offspring you can make with the best genes, is really going to make you fit for your environment, make your lineage live for a long time. You also have things like genetic drift. You have migration. And a big part is mutations. So mutations are errors in the DNA code. So there are errors in the DNA code, and that can bring, since DNA is going to affect the amino acids that are put together, affect the proteins, that affects the trait. So if there's a change in the DNA, that could lead to a change in a trait. And if it's bad, it's not going to help them. But if that mutation helps, they're going to live longer, reproduce more, pass those on to their offspring, and the offspring are going to have that trait as well. And then they can do the same. They can pass that on if it helps them in their environment. So when we talk about evolution, it's not necessarily the idea of being able to live, but the idea that you're able to reproduce. That's why we're here. That's why an organism is here, is being able to produce and pass on their genes. If you are an organism, you're saying, hey, I'm here for a reason. I'm here because I have great genes. And what I want to do is I want to pass on my genes, pass on my traits to the next generation. So if you are not able to do so, if you don't live long enough, if you're not fit for your environment to reproduce, then we don't say that you are a fit organism. So you could live a very long time, but if you don't reproduce, or there's something with your traits that stops you from reproducing, then that does not mean that you are fit for the environment, just in evolutionary terms. So keep these things in mind. If you do have questions, please contact me so I can clarify anything. But this is really the bulk of the final. You need to understand these concepts and these ideas. Now, the nitty gritty stuff, probably not going to see that, but these general ideas, you must know them.